Okay, so welcome back to this next video in which we are discussing lipoproteins and the transportation of lipids around the bloodstream. Okay, right, so in this next video what we now want to talk about is where these chylomicrons are going to go. Okay, so at the moment we've just talked about how the chylomicrons have been released from these enterocytes, which are these simple columnar epithelial cells that line the surface of the small intestine, and they have gone into the central lacteals. Okay, now where does the lymphatics of the intestines go? Well, basically all of the lymphatic vessels all for the intestine goes to something known as the Cisterna Chyli. Okay, so let me just show you the anatomy of the Cisterna Chyli. So basically we're going to look at the lumbar vertebral level. Okay, so these are the centrums of the vertebra basically. Okay, so we have L1's vertebral body here, we have L2's vertebral body here, and then we have the vertebral body of L3 here. So basically we're just looking at a little portion of the vertebral column. Above here will be the thoracic portion of the vertebral column, and below here you'll then have the sacrum. Well, you'll have L4 and L5, and then finally underneath that the sacrum and the pelvis attached to the sacrum. Okay, right, so basically you have the Cisterna Chyli at the level of L2, and it is a large sac of lymph basically that is formed by two major lymphatic vessels joining together. Okay, and these are the two lymphatic vessels which drain the two lower limbs. Okay, these are called the lumbar trunks, and you have a left lumbar trunk and then a right lumbar trunk. The left lumbar trunk will supply the left lower limb, and the right lumbar trunk here will supply the right lower limb, and of course we're talking about patient's left and patient's right. Then they coalesce basically to form a big sac, and this big sac is called the Cisterna Chyli. And basically all of the lymph from the intestine also drains into the Cisterna Chyli, which sits just in front of the vertebral body of the L2 lumbar vertebra. Okay, and then what's going to happen is you have a very large lymph vessel draining the Cisterna Chyli, and this large lymph vessel that drains the Cisterna Chyli is called the thoracic duct. Okay, and this thoracic duct is now going to make its way up the vertebral column. So it's going to go up through the thoracic vertebral region, and it's going to eventually drain into the left subclavian vein. So let me show you the anatomy then of the great veins. So here is the superior vena cava. Okay, and this will go into the right atrium. So the superior vena cava is usually just abbreviated to the SVC. So S stands for superior, V stands for vena, and then the C stands for cava. Okay, and this will be going to the right atrium of the heart. Okay, so this is the downwards portion where it will be joining the right atrium here, and here is the superior portion up here, which is going to be formed by the joining of two large veins. Okay, and these two large veins which join to form the superior vena cava are called the brachiocephalic veins. Okay, so we're looking at the left side, so this is the left brachiocephalic vein. So we're imagining that we are looking from the front of the person at their large veins. So this is the person's left side. So this is the left brachiocephalic vein. And even the left brachiocephalic vein itself is made by another two veins joining together. Okay, and these two big veins that join together to form the brachiocephalic veins, and it's the same on the other side, I'll just draw it here. Okay, these two big veins that join together to make the brachiocephalic veins are the subclavian vein. Okay, so this one here is the left subclavian vein. Okay, whilst this one over here is the right subclavian vein, so this is the left subclavian vein, and now this vein that comes down from the head, this is the internal jugular vein. So again, this on this side, this is the left internal jugular vein, but of course you also have on the opposite side the mirror image of this, which is the right internal jugular vein. Okay, right, so basically the thoracic duct, initially, as I've shown here, it's in the midline. However, as it gradually ascends, it moves over towards the left side, and basically it's going to come up like so. So this is the thoracic duct coming up here, and it's going to go behind the brachiocephalic vein here. So it's behind this left brachiocephalic vein. It then goes behind the left internal jugular vein. In fact, I might show dashed lines. In fact, no, I'll just show it. But remember, it's going behind these because it's just too difficult to show it without actually drawing it. Okay, and then it's going to join on to the left subclavian vein just before the left subclavian vein joins with the left internal jugular vein, basically to form the left brachiocephalic vein. So shaded now in turquoise here, this is how the thoracic duct joins the left subclavian vein. So basically it is asymmetric. It starts off right in the middle, but then sort of slants over to the left-hand side and then drains into the left subclavian vein like so. Okay, right. There is a right side equivalent which does not drain the Cisterna Chyli. Basically, the right side equivalent just drains the lymphatic vessels at the right thorax and then drains into the right subclavian vein. Okay, but it doesn't make it all the way down to actually drain the Cisterna Chyli, and that right side equivalent is just called the right lymphatic duct. Okay, right, so this is the thoracic duct, the one and only thoracic duct, and it sort of leans over onto the left-hand side. So basically, all the chylomicrons that we are putting into these lacteals, they will eventually drain into the Cisterna Chyli here, and the Cisterna Chyli will drain into the thoracic duct here, and the thoracic duct will then drain into the blood. So indirectly, yes, but nevertheless, these chylomicrons do end up in the blood. So we now want to see, once we've got these chylomicrons into the blood, where are they going to go? Okay, so at the moment they are absolutely bursting full of triacyl glycerols. So basically we are going to send these to tissues that can actually either use or store triacyl glycerols. So we're going to send them to adipose tissue and also to muscle tissue, such as skeletal muscle tissue, and also the heart. The heart absolutely loves fat molecules, specifically triacyl glycerol molecules. The heart and also skeletal muscle actually prefer to use fat molecules than carbohydrate molecules. Contrast that to a tissue like the brain, for instance, which absolutely refuses to use fat molecules and will only use glucose molecules, or ketone bodies is the little exception that it will make. Okay, right. Now, just before we go any further, what I would like to discuss is going back to these bile salts, okay, wherever they were. So these bile salts, remember, formed these mixed micelles, and basically these mixed micelles then sort of went down the gaps between these microvilli and passed the products of digestion into the enterocyte, where they then went off to the smooth endoplasmic reticulum to be reconverted back into substantial lipid molecules. Okay, but these mixed micelles actually are recycled over and over again, so they don't get absorbed themselves. The bile acids are not going to be absorbed instantly by the enterocytes. Instead, the micelle that is formed by the bile acids, that will go back out now, and it will go back into the luminal contents, and it will collect up more digestion products and then come back. Okay, so it's recycled over and over again. We use them multiple times in the same digestion session, if you like. Now, You might then ask, well, are they just going to stay in the contents of the lumen and be excreted out? Well, the answer to that is no. We try our best to eventually reabsorb all of the things that we put into the bile. So the lecithin molecules that we put into the bile, those are all being reabsorbed, because remember, they're being digested into lysophosphatidylcholine and then a free fatty acid, and those are being passed into the enterocytes. So we've discussed how we reabsorb the lecithin of the bile, but how do we reabsorb the bile acids? Because I'm just told you they're not really being absorbed, they're staying in the lumen and continuing to perform their ferrying action of digestion products to the enterocytes. So how do we reabsorb the bile acids? Well, basically, reabsorption of the bile acids doesn't occur until the final portion of the intestine. So right in the ileum, finally, you will reabsorb the bile acids, and you'll want to reabsorb as many as possible, and these go straight into the blood, so they don't go into the lacteals, they don't go into chylomicrons, they go directly into the blood. Okay, right, and then they will go back with the portal blood to the liver, and the liver will then take them back out, and then it can repackage them into bile. And that concept of the bile acids being secreted by the bile duct into the intestine at the level of the duodenum, and then being reabsorbed into the portal blood at the level of the ileum, and then going back to the liver, that concept is called the enterohepatic circulation. Okay, now of course it's not perfect. There are always some bile acids which won't be reabsorbed, but most of the bile acids will be reabsorbed. Okay, so that concept of bile acids circulating between the liver and the intestine is known as the enterohepatic circulation. Okay, so entero means pertaining to the intestine, hepatic means pertaining to the liver, so this is the circulation between the intestine and the liver. Okay, right, so now let's move on and discuss chylomicrons. Okay, so we've covered digestion of fat now. We've now got these chylomicrons within our blood, and we want to see where are they actually going to go. So basically they're going to go to adipose tissue and also to myocytes. So basically in the capillaries of adipose tissue and myocytes, the endothelial cells have the necessary enzymes to break down the contents of the chylomicrons. So let me draw this. So I'll draw the structure of a capillary. So capillaries are tiny little blood vessels. They are barely 1 cell thick. Okay, so their walls are extremely simple. You have a layer of endothelial cells, shown here. These are the endothelial cells. Here are their nuclei. And these endothelial cells are sitting on a basement membrane. Okay, so the basement membrane is a protein meshwork which is quite rigid and therefore supports the endothelial cells nicely and holds them in place and in shape. Okay, so this is the structure of the wall then of a capillary. You have literally only an endothelial cell sitting on a basement membrane, and that is it. You do have little cells occasionally dotted around the outside called pericytes, but we won't worry about them. Okay, so in capillaries which go through adipose tissue and also muscle tissue, okay, and we're talking about either skeletal muscle tissue or cardiac muscle tissue rather than smooth muscle tissue, so either skeletal or cardiac myocytes. Basically, in the capillaries which go through these forms of tissue, you will have endothelial cells which have the equipment on their apical surface to harness the fatty nutrients within one of these chylomicrons. Okay, so what apparatus do you need on your surface in order to access the fatty contents of a chylomicron? Well, basically, you need something called lipoprotein lipase. Okay, and your lipoprotein lipase is usually abbreviated to LPL for short. So the first L stands for lipo, The P then stands for protein. Okay, so that makes sense. Since we're talking about a lipoprotein, we're talking about chylomicrons. And then lipase is the second L, which just means something which is capable of hydrolyzing these ester links between long-chain carboxylic acids and the alcohol groups of glycerol. Okay, right. So, if we draw an endothelial cell now bigger, okay, so let's let this represent one of these endothelial cells which lines capillaries within adipose tissue and also myocytes. Then basically on the apical surface of the endothelial cell, which means the face which faces into the lumen, you have a certain molecule which is present as a dimer, which holds on to lipoprotein lipase. Okay, so here are these two molecules, and basically these molecules are what are known as GPI anchored high-density lipoprotein binding protein 1. Okay, so GPI anchored, and we'll discuss what GPI anchored actually means in a moment. So they are GPI anchored, and then high-density lipoprotein, which I'll just abbreviate to HDL, binding protein 1. So these two proteins that you have on the apical surface of your endothelial cells within adipose and also skeletal and cardiac muscle tissue have these proteins called GPI-anchored HDL binding protein 1s. Okay, and for short, the GPI-anchored HDL binding protein 1 is usually abbreviated to GPI, okay, then H for HDL, and then BP for binding protein, and then 1. So GPI-HBP1 stands for the GPI-anchored HDL binding protein 1. Now, these proteins do not have transmembrane domains. Instead, they are attached to the outer surface of the lipid bilayer of this endothelial cell by a GPI molecule. So let's now talk about what a GPI molecule is. So GPI stands for glycosyl phosphatidyl inositol. Okay, so this shouldn't take us too long to work out what this is, because we know exactly what phosphatidyl means. That refers to a phosphatidic acid molecule. So this is glycosyl phosphatidyl inositol, and it's all one massive great big word. So basically what this molecule is it is a phosphatidic acid molecule. Okay, so we can start off with that. That's the phosphatidyl bit here. So I'll underline that in red. So this phosphatidyl bit refers to the fact that the core structure of this molecule is a phosphatidic acid molecule. Okay, so here we have our phosphatidic acid. We have these two long-chain carboxylic acids esterified to the first and second alcohol groups of our glycerol molecule here in green. And then to the third alcohol group of the glycerol molecule, we then have a phosphate group linked via a phosphoester link. So that's phosphatidic acid. We want to add on to the phosphatidyl group an inositol molecule. Okay, so we are going to stick onto this phosphate group via a phosphoester link another molecule called inositol. Now, inositol is again the old biochemist's name for a molecule that is now more correctly called cyclohexane 1,2,3,4,5,6 hexol. Okay, so the proper name for inositol is cyclohexane 1,2,3,4,5,6 and then hexol. Okay, and although again cyclohexane 1,2,3,4,5,6 hexol is a complete mouthful, it's a useful name because it tells us exactly what we're dealing with. Cyclohexane tells us that we are dealing with a 6-membered carbon ring where all of the bonds between the carbons in this ring are single bonds. Okay, the 1,2,3,4,5,6 hexol tells us that every single one of these carbons has an alcohol group coming off, meaning that overall the entire inositol structure has 6 alcohol groups coming off, one off every single carbon atom of the inositol ring. Okay, and then to make up the final bond that every single carbon needs, you'll also have another hydrogen atom. Okay, so basically what you have done is you have taken this inositol ring here, and you have bound the alcohol group of this carbon here to the phosphate group of the phosphatidic acid molecule, and that creates you phosphatidyl inositol. Now, to turn phosphatidyl inositol into glycosyl phosphatidyl inositol, you have to link certain sugar molecules onto this inositol ring. Glycosyl, anything with glyco in, means sugar. Okay, so we are going to add in now a few sugar molecules. Specifically, we're going to add in 4 sugar molecules. 1, then another one here, 2, a third one here, and then a final 4th one here. Right, so what sugar molecules are these? Well, basically, you start with a glucosamine here. So this first one is what's known as a glucosamine molecule, and then the next 3 are all mannose molecules. So you have mannose after mannose, and then a final mannose. Okay, right, so those are the 4 carbohydrate molecules that you have within a glycosyl phosphatidyl inositol group. So I'll colour these in. So we'll have glucosamine here in turquoise. Okay, we'll then have in pink the 3 mannoses. So 1, 2, 3, 4. Sorry, 1, 2, 3. Okay, right. Now, basically, we can attach this group onto a carboxylic acid terminus of a protein. So it's a common lipid modification. It's one of the common lipid modifications that you see in a lot of proteins. So lots of different proteins can have glycosyl phosphatidyl inositol molecules linked onto them. Now, we haven't quite finished the glycosyl phosphatidyl inositol yet. It has one final little bit added onto it, which is a phosphoethanolamine molecule. Okay, so basically, off this mannose here, you're going to have a phosphate group coming off here. So this little ball here is supposed to represent a phosphate group. And then attached via a phosphoester link to this phosphate group, you then have a molecule known as ethanolamine. And basically, this has a very similar structure to choline. Okay, so you have a 2-carbon structure, and then you have an alcohol group at one end, which is involved in the phosphoester link to this phosphate group here, and then on the other end you just have an amino group, like so. Okay, and that now is the complete glycosyl phosphatidyl inositol molecule. Okay, so this final group that we've got here, which is this ethanolamine with a phosphate group, is called a phosphoethanolamine molecule. Okay, so this is phosphoethanolamine. And basically we can now add this molecule onto proteins. So this is a common lipid modification. So remember, proteins fundamentally are polymers of amino acids. Okay, so you have an amino terminus at one end, which is where the first amino acid has no carboxylic acid of the previous amino acid to bind to. And then you have a carboxylic acid terminus at the other end, which is where the final amino acid has no amino terminus of the next amino acid along to bind to. Basically, what you can do is you can bind this amino group of the glycosyl phosphatidyl inositol molecule to this carboxylic acid group at the terminal end of the polypeptide, okay, and that will latch this entire GPI group here onto your polypeptide. And that's a common lipid modification that can occur to proteins. Now, these glycosyl phosphatidyl inositol molecules implant into the phospholipid bilayer, generally the outer layer of the phospholipid bilayer of the cell membrane. So their two long-chain carboxylic acids here hold them within the phospholipid bilayer, and that then latches the protein onto the outer surface of the cell membrane, and that is how these proteins are being held at the outer surface of the cell membrane of these endothelial cells. They are attached to these GPI molecules which are anchoring them at the outer surface of the phospholipid bilayer. And then what happens is you get two of these, they bind to one another, and they're now dimerized. Okay, now what can happen is these GPI-anchored HDL binding protein 1s here can now bind to a homodimer of lipoprotein lipase. Okay, so on top of this, what you're going to have is a dimer of lipoprotein lipase. So basically, in order to function, lipoprotein lipase needs to be dimerized like this. And let me explain why. So basically, if we just have a little look at the structure of lipoprotein lipase, and don't worry, I'm not going to give you any sort of residue by residue analysis, because I don't think that's actually known for lipoprotein lipase. I don't think we even have a crystal structure yet. Okay, so all that is known is that you can split the protein roughly into two different parts. Okay, so one part is called the amino terminal domain here. So this is the part near the amino terminus, and then the other part is called the carboxylic acid terminal domain. Now basically, these are the two halves of the enzyme, so for the enzyme to function you need both of these, okay, but it's difficult for this one to interact with this one when these two are on the same protein. So basically what you need to do is get another one of these things and put it the other way round, okay, like this, so, and this will make a lot more sense when I've added some colour onto this picture. Okay, so that the carboxylic acid domain of this second one down here, which I'll colour in pink, can now interact with the amino terminal domain of this first one. Okay, so the amino terminal domains are in green, the carboxylic acid terminal domains are in pink. So this basically forms a functional enzyme here, and then this one will also form a functional enzyme. So you have to have them dimerized for them to work, but you do actually have two functional enzymes in that homodimer. Okay, so this is called a lipoprotein lipase homodimer because the two proteins that are involved in the dimer are identical. So homo means same. Okay, so basically, on the endothelial cells of capillaries within adipose tissue and also within skeletal muscle tissue and also cardiac muscle tissue, the endothelial cells have this assembly on their surface. And basically, this assembly can harness lipid molecules that are within chylomicrons. Okay, so what's now basically going to happen is this thing is going to stick itself into the chylomicron, okay, like so. So here's our chylomicron, and basically let's let this represent the outer monolayer of phospholipids, okay. What will happen is the LPL molecules, the lipoprotein lipase, will stick itself through the phospholipid monolayer of the chylomicron. So here is our chylomicron. And now basically it has access to the goodies inside. Now one of the key goodies that you have inside of these chylomicrons is these triacyl glycerols. Okay, so here is the glycerol molecule in green, and here are the three long-chain carboxylic acids in orange. Okay, now what can happen is the lipoprotein lipase can now start to break down these triacyl glycerols. But I should just mention that in order to activate the lipoprotein lipase, you need apoprotein C2 to bind. So remember, in the phospholipid monolayer on the outside of these chylomicrons, you have a great many different apolipoproteins. So for instance, we discussed apolipoprotein B48, apolipoprotein C2, apolipoprotein E, and then there were those other more niche ones, apolipoprotein C3 and apolipoprotein A4. One of the key ones is this apolipoprotein 2, and I will colour this in. I think I'll try yellow maybe. I might regret this though. It's bound to smudge. Okay, so in yellow here are two molecules of apolipoprotein C2, and you can see that they are binding with the lipoprotein lipase. Okay, so in yellow here, this is apolipoprotein C2. And when the apolipoprotein C2 binds to the lipoprotein lipase, that will activate the lipoprotein lipase. And now the lipoprotein lipase is going to set to work on these triacylglycerols here. Now what's it going to do? Well, it's going to do the same reaction as pancreatic lipase. Okay? So basically, it's going to take the triacylglycerol molecules here, and it's going to break them down into a 2-monoacylglycerol and also 3 fatty acid molecules. Okay, right, so here is the 2-monoacylglycerol, and here are the 2 free fatty acid molecules. Now, this 2-monoacylglycerol molecule, this will remain in the center of the chylomicron. But the free fatty acids, those are going to get out of the chylomicron. They will leave the chylomicron and they will diffuse across the endothelial cells, across the basement membrane, and to the peripheral tissue. Okay, so they will go either to the adipocytes or the skeletal or cardiac myocytes. Okay, now if you are a adipocyte, what will happen is you will take up these free fatty acid molecules. So let's draw an adipocyte here. Okay, so adipocytes, remember, are these cells which have a massive, great storage vesicle full of lipids. Okay, and what will happen is these free fatty acid molecules, they will come into the adipocyte, and the adipocyte will reconvert the free fatty acid molecules into triglycerides, and they'll then store these triglycerides within their huge storage vesicle here. So here's the glycerol molecule in green, and then here are 3 long-chain carboxylic acids esterified to that glycerol molecule. And this is supposed to represent the nucleus of the adipocyte. So this is an adipocyte. Okay, right. So adipocytes will store the triacylglycerols then. Okay, now if you are a myocyte, so I'll draw a myocyte here, you're not going to store the lipid molecules, instead you're going to burn them. Okay, so you are going to use them for energy, and the heart is absolutely fantastic at doing this. It loves free fatty acid molecules to produce ATP. So basically, you will send it down a process called beta oxidation, and this will result in the production of acetyl coenzyme A, which can then go through the Krebs cycle to generate reduced nicotinamide adenine dinucleotide, which can then go through the electron transport chain to produce that proton gradient across the inner membrane of the mitochondria, and then ATP synthase will release the protons, capture the energy, and use it to bind ADP to inorganic phosphate to create ATP. Okay, so basically the myocytes use the fat molecules for beta oxidation. Okay, so the chylomicrons then are going to go round, they're going to go around the circulation, and they will come into contact occasionally with these endothelial cells of capillaries that are within adipose tissue or myocyte tissue. Okay, and when they come into contact with that, they will get their triacylglycerol content completely depleted. However, the important thing to note is that the adipocytes are not going to we're not going to remove other components of the chylomicrons. Okay, so So we're not going to remove, for instance, the phospholipid monolayer, we're not really removing the apolipoproteins, and we're not really removing the cholesterol or the cholesterol esters. We are just taking the triacylglycerols, okay? And this is going to result in the production, basically, of chylomicrons, which don't then have that much triacylglycerol within them, but they have a lot of cholesterol within them, a lot of cholesterol esters, and they still have their phospholipid monolayer and their apolipoproteins. Okay, so what are these structures called, and what's going to happen to them? Okay, well, basically, they are called chylomicron remnants. So when you've got a chylomicron which doesn't have that much triacylglycerols left within it, okay, because it's given them all up to peripheral tissues such as the adipocytes or the myocytes, then it's called a chylomicron remnant. Okay, now what's going to happen is that this is going to go to the liver, basically. So now the chylomicrons will go to capillaries within the liver, and they will be removed from the blood by the hepatocytes. So let's draw a hepatocyte here. So basically, the hepatocytes will take the chylomicrons out of the blood, and what they're going to get from removing the chylomicrons from the blood is they're going to firstly get a nice supply of phospholipids, which will mainly be phosphatidylcholine, and we know that liver cells love phosphatidylcholine. They can, chuck that straight into the bile. Okay, you're also going to get a whole bunch of cholesterol esters. Now, the instant the cholesterol esters arrive at the hepatocyte, they will be deesterified to just cholesterol. Okay, you're also going to get some cholesterol arriving from the chylomicron, and then finally, you've also got some of the 2-monoacylglycerols arriving as well. So this is what the hepatocytes will receive from the chylomicron remnants. Now they can use the cholesterol that they are producing here that they're getting from these chylomicrons to produce new bile acids, because remember, some of the bile acids will not make it back into the portal blood. Instead, they will be secreted within the feces, so we're continuously losing bile acids. So the hepatocytes are going to have to continuously be producing more bile acids. Okay, so we'll call it there for this video, and in the next video we'll start discussing VLDL.